Hello everyone, so this is Shruti Ahuja and in this video we are going to talk about glands in histology. So it is a very short topic and usually it comes as a 5-mark question in your theory exams. But if you study it alone with histology purpose, then it will also help you when you will study GIT histology, respiratory histology and much more, because it is a kind of foundation topic which will help you further, right? So basically we can say it is a part of general histology. So glands are of two types, exocrine and endocrine. As we all know, exocrine is into a duct and endocrine is D. So it is directly into circulation, right? Therefore, rich capillary network is required for endocrine glands. So in endocrine, we are having individual cells, endocrine tissue in mixed glands and separate endocrine organs. In individual cells, we are having as in digestive organs, enteroendocrine cells, and in the GIT tract, we are having cells as a gland. Now endocrine tissue in mixed glands like pancreas is a tissue, we say, right? And separate organs is like pituitary, thyroid, parathyroid, which are solely endocrine glands. Why pancreas is here and not here? Because it has some endocrine tissue plus exocrine tissue, which makes it a compound gland, right? Now exocrine into duct. So it is basically unicellular and multicellular, right? Gland type. So in unicellular, we are having goblet cells. We are having these cells in the GIT. And in multicellular, we are having there is one secretory portion and there is one ductal portion, right? So if we say multicellular, they are of two types, simple, or we can say simple means no branching is there, right? And branched is compound. So simple unbranched is straight. So I can show you something like here. Here you can see, first of all see this, and now you can appreciate there are curvings here. So it is basically straight. This is the surface epithelium and these are the secretory cells. So unbranched simple tubular exocrine glands. Example is this. These are intestinal glands, right? Now if we say it is straight, and we are also having the now branched. In simple unbranched, we can have these type of straight, and we can also have something coiling is also there, right? They can be rounded and coiled structures. Now branched, in branch we are having compound are tubular, tubulo-acinar and acinar, right? So in tubular, it is this is the surface epithelium and these are secretory cells. So Y-shaped structure is you can appreciate, right? So here also you can see this thing is going like this and it is being divided here. If you compare these two, this is straight like a tubules, and here it is being divided, right? So this is the tubular and it is branched with tubular. Now in acinar, you can see there are so many rounded structures. There is it is very hard to see any tubule here, but this is this is acinar. Now in tubulo-acinar, here you can see the tubules, right? And here you can see the acini as well. So we can say this is tubulo-acinar. The examples you will learn later on, but here it is important to understand the types and morphology of these glands. So other classification is mucus, serous, mixed. This classification is important in the case of salivary glands, right? So next is merocrine, holocrine and apocrine. So merocrine is exocytosis like pancreas, holocrine the whole cell, whole cell. Skin, sebaceous glands, and apocrine is mammary glands, in which it is a pinched off type, right? And now we say apocrine is under merocrine, right? So this was all about the glands, and it comes as a 5-mark question usually in general anat paper or in your final exam, less commonly. But if something comes from general anat, it is we can say the question of choice. in some cases. So if you like this video, then do like it, share it, and also do share your doubts in the comment section.